can you just give a brief overview of what your study and what the findings were and the importance for clinicians?
So our study focuses on using single nucleus RNA sequencing to compare genetic expression profiles from autopsy brain samples from individuals with Alzheimer's disease with either European or African local ancestry who are carriers of the homozygous APOE4 gene. Our study is important to clinicians as the therapeutic question concerning APOE4 has fallen into two possibilities. One, that the risk from APOE4 for Alzheimer's disease could be due to this gene being inefficient or unable to produce their normal function. And if that is the case, then increasing the expression of APOE4 will be therapeutic. On the other hand, if the cause is that the APOE4 gene is the one causing the problem, then the opposite approach will be indicated, which would be reducing the expression of APOE4 and for that to be therapeutic. Our latest findings suggest that the reduction of APOE4 expression will most likely be the therapeutic choice.
How does this generic risk factor lead to potentially higher risk of Alzheimer's for some populations?
So we have previously shown that the difference in Alzheimer's disease risk from APOE4 carriers is associated to the local ancestry region. In other words, to the specific genetic region surrounding the APOE4 on chromosome 19. In that study, we found that if an individual inherits the APOE4 allele from an African ancestor, they have a reduced risk compared to the Europeans and Japanese. What the protective effect or the protective genetic factor is in this case, we don't know. And that's the main reason why we wanted to do this study. We do know that the structure of the APOE4 protein is not different from different ancestries. So we believe that the difference relies on how either the APOE gene or other genes within the region are regulated between ancestry. Our latest finding suggesting that European carriers have increased APOE4 expression compared to Africans fits within this hypothesis.